Good morning, everyone. Hope everybody's awake and ready on a Monday morning here in February. It's an exciting morning, actually, when you think about what the Minneapolis Heart Institute Foundation is all about. So I stand in front of you as a proud board member of the foundation and excited to introduce Dr. Felipe Kazmirczak today to give a really interesting talk, but also stand in front of you also as a physician leader of the practice and somebody that gets the joy of recruiting great physicians to our practice. And I think I can say without a doubt that Felipe is here because of the foundation, but also because he's a phenomenal cardiologist. And so super excited to kind of combine both of those this morning and hear from Felipe. Felipe joined us last summer, last July from the University of Minnesota. He actually earned his MD from Lutheran University in Brazil and completed his internal medicine residency at the University of California, San Diego. He moved then to Minneapolis and Minnesota to join the University of Minnesota, where he did his advanced imaging fellowship as well as his general cardiology fellowship at the University of Minnesota and worked as a faculty there until July of 2024 when he came to join us. And so we're thrilled that Felipe is here for sure. His interests include integrating advanced cardiac imaging with translational science to understand how pathogenic pathways can be targeted for pulmonary hypertension and right heart failure, and that's what we'll hear about today. His work has actually been recognized already as a Young Investigator Award finalist at the ACC in 2023. He is published in Circulation Research and has a recent publication in the Blue Journal. Today he'll talk about his work regarding the effects of intermittent fasting on cardiac function and how it got him into the path of studying ferroptosis. So Felipe, thanks for joining.
Absolutely. Thanks so much for this. Thanks for this kind introduction and the opportunity to present here at the Minneapolis Heart Institute. So today I'll take you on a journey that for me started about 20 years ago. And that's when I was in med school. I started learning about cardiac metabolism. I was fascinated by it. Over time and during my training in cardiology, I was fortunate to work with a group of very talented people with different skills that was able to allow me to develop my passion and work on several different ways to understand cardiac metabolism function. And today I'll share with you the work that we have done over the past 5 years with this group. Regarding disclosures, I'm co-investigator in R01, which we're looking for inflammatory modulators for changing in right ventricular function. For today's objectives, we're going to be talking about cardiac metabolism, just a brief overview so we understand things more easily. And then we're going to pivot into intermittent fasting, its relationship to the right ventricular function. Then we're going to move into this phenomenon called ferroptosis and inflammation-related pulmonary hypertension. Later on, we're going to talk about target therapies for right ventricular failure. So this concept fascinated me, which was that the heart, as it starts to fail, become an energy is starved out of you. So it's like when the heart needs the most amount of contractility, mechanical work in heart failure, that's when it starts to starve because there's either lack of fuel availability, or ability to burn. And this is not a new concept. This was published in 1939 by Herman et al. And this paper in The Chemical Nature of Heart Failure, which is very elegant observation, which was heart failure hearts had lower phosphate, meaning highly energetic phosphates compared to normal hearts. If you think about phosphates, the easy way to remember is ADP, ATP. ATP is a 3-phosphate, ADP is lower. So having more phosphate means that you have more electrical energy to perform mechanical work. Fast forward to 2007 New England Journal. That's the engine out of fuel title. And we're just going to dig in a little bit into the myocardial energetics. And the way I like to break it down is in two parts. One, we have a fuel source. The question about the fuel source is, one, is it available and can we use it? So the preferred fuel source for the heart is fatty acids, as you can see here. And fatty acid needs to be transported into the cell. And then within the cell, you need to use those fatty acids. That happens through beta oxidation. Then we get into the Krebs cycle. So if you remember from med school, that's when we get to the mitochondria, which is going to do the second phase which is burning that fuel and creating energy. So within the mitochondria, you're going to have respiratory chain complexes that are going to allow to create an electrical gradient. That electrical gradient is going to be what's going to drive this pump into cycling ADP, adding a third phosphate, creating ATP. And ATP is going to be used by the heart. So when we look at the heart uses a lot of energy to perform mechanical work. Like we talked about, the preferred fuel source for the heart's fatty acid oxidation. As heart failure develops, for two phenomena, the heart's not able to utilize this preferred fuel source. And that happens for one, it's harder for the fuel to get to the heart or gets stuck inside the cell with some deleterious effects. And second, the burning areas of the heart that create the energy, mitochondria, starts to become dysfunctional. And we're going to talk about that too. Overall, what happens over time as you develop heart failure and decompensated heart failure is that your availability of fatty acid utilization goes down. The heart tries to compensate that, and that's increasing glucose metabolism. Glucose metabolism, unfortunately, compensates initially, but as a lot of things that we know about heart failure, that initial compensation on the long term, it's deleterious. And what happens is with glucose metabolism, you have a lot of reactive oxygen species, meaning a lot of stress in the cell. That leads to a couple of proteins that lead to less efficiency overall in the cell. And in the end, the heart cannot even glucose utilization anymore. And I think that's, looking at this bioenergetics, one of the reasons that we're seeing now medications that target energy metabolism being shown to work in heart failure, like GLP-1s and SGLT2 inhibitors. And I think there's a lot to come in this modulation of energy and ability to use energy in the heart. Most of that work was observing the left ventricle, bigger cavity, and we're left ventricular centric. But what about the right side of the heart? So on the right side of the heart, we have shown that as you develop pulmonary arterial hypertension, you increase the load on the right ventricle. You have a signal to bring more fuel in the way of triglycerides or free fatty acids. Unfortunately, because you're not able to burn that fuel starts to get accumulated. One easier comparison, the way that fuel gets accumulated in the heart, is how fat gets accumulated in the liver, steatosis, NASH. So similar process is just not an efficient way that creates a lot of oxidation, that creates a lot of reduction on your metabolic capacity. And that has been shown by Britain that increases ceramides and other components and overall decreases mitochondria function and ability to oxidize, reducing cardiac function overall. And why RV failure matters. The RV failure is the leading cause of death in patients with pulmonary arterial hypertension. In multiple cardiac states, having heart failure from the right side is an independent predictor of mortality. And it's been shown in several studies when you use ways of quantifying right ventricular function that the mortality is significantly higher in patients with right ventricular failure. And also when you look at hospitalization-free survival, patients with right ventricular failure have a lot lower of hospitalization-free survival. So this is a major clinical problem. The right ventricle, unfortunately, there is not a target therapy. For the left ventricle, we have several therapies available: ARNI, ACE, ARB, beta blockers, you name it. But for the right ventricle, there is not a single therapy that it is target and has been proved to work. And that could be for multiple reasons. The left ventricle is very different than the right: pressure, load, wall thickness, nerve system, embryology. So we just need to understand that and trying to find a way to get a treatment that works on the right side of the heart. Based on the importance of the right side failure and this idea of energy modulation, we decided to look into an intervention called intermittent fasting. So what intermittent fasting has shown across multiple studies is that it modulates energy and increases what we're looking for here, health span, meaning RV failure, pulmonary hypertension, chronic diseases, and they happen over the course of the life. So the longer you can expand your health span, meaning the years you don't have chronic disease, we would think that would modulate and reduce the intensity of this chronic disease. And that's the one thing that intermittent fasting does. There's other studies showing the caloric restriction in populations about 800 to 900 calories per day also has similar benefits over time. And if you look at the blue zones and the longevity people, they tend to eat a lot lower caloric diet, but also a lot less nutrient-rich diet in a way, less processed. When we look at lifespan, we're talking about health span before, now lifespan. Intermittent fasting is one of the few interventions that has been shown across multiple species to prolong health span. And that goes from bacteria, that goes through yeast, worm, and mice. And this is from Valtteri Longo. He publishes a lot in longevity, healthy aging, and fasting, including fasting mimicking diet. So it's a great source to look for if you have some interest in this area. So we talk a lot about intermittent fasting, but what is intermittent fasting? So intermittent fasting, breaking it down in simple terms, works in two main ways. One, you take away the nutrients, in this case, the protein, carbohydrates, and fat, and you're going to generate several metabolic adaptations to do what we call metabolic switching. So that is, you have one type of fuel that you're burning, mostly glucose for us that are eating. And then as you start to fast, you're going to start to burn more free fatty acids. And then as you keep fasting, you're going to start burn more ketones. And that metabolic switching is a way to train your cells for this small amount of stress, but to allow the cell to be more resilient. The other way that works is through neuroendocrine pathways. And one of the biggest pathways that get activated is, as you stop eating, that's going to be one of the few ways you're going to have a low blood sugar with low insulin. Most of the time for us to get a low blood sugar, you need to give more insulin. In this case, you're going to have very low insulin. And insulin is a build hormone. It's an anabolic, create cells, keep them. When you take away insulin, you allow a lot of the repair processes to start happening. And that's another neuroendocrine pathway that's used by intermittent fasting. Glucagon also goes up, so you can create gluconeogenesis, among others. And all of those factors combined are going to lead to stress resistance. As you're fasting, you took away the insulin, which is a build hormone, and then you added mTOR, you activate autophagy, meaning you go around, see the cells are now working well, and then you go into control cell death. Glucose metabolism and lipid metabolism also gets improved. And then one of the biggest factors is, as you create autophagy for those cells that are not able to create energy, the organelles, in this case mitochondria, as soon as you eat, you break your fast, you send a signal to create brand new mitochondria, which by definition should be more efficient, and that's called mitochondrial biogenesis. The overall goal is to stress the cell just like we stress ourselves when we do exercise, when we move around, so then we improve our health span and our longevity. When we look at intermittent fasting and we break it down, like how does it really work? And the easier way to think about it here, we're going to talk about this central element here, which is called AMPK. And the way to think about AMPK is just like an energy sensor. So as soon as you take away energy, that energy sensor is going to realize that and start activating. So the more you take, meaning the longer your fast is, more activation you're going to have. Or if you exercise because you're burning more energy, same thing. So the more you exercise, meaning if you run a marathon, huge activation of this AMPK. Caloric restriction, similar. And AMPK is a major regulator of cell metabolism. So the things we're interested from AMPK here is mitochondrial biogenesis. So in heart failure, we can burn that fuel. Beta oxidation, so we have the fuel available for us to burn. Fatty acid synthesis and metabolism. And then also autophagy, so we can take away some of the cells that are not working as efficiently as they should be. So based on this background, the importance of right ventricular failure and the bioenergetics, we set out to do a study which was intermittent fasting as our intervention to look to if we can prevent those maladaptive metabolic changes. And we're going to use this AMPK sensor as our surrogate to see if we're having an effect or not. So our work was initially done on a pulmonary hypertension model. So this model is a rodent. You inject motocrotaline, which is an sclerotic agent. So that creates a sclerosis in the pulmonary vasculature. That sclerosis increases your pulmonary vascular resistance, pulmonary hypertension. As increases your pulmonary resistance, pulmonary hypertension, you have RV after load increase, right ventricular failure. So that's what this model is. And what we did was we had a control. We had an ad lib that food was available and the rodents could eat as much as frequently as they wanted. And then we had an intermittent fasting that would eat every other day for about 3.5 weeks. This is we submit this initially, and then we got crushed on the way that this is a very inflammatory model. When you sclerose a vasculature, you're going to generate a lot of inflammation. So the criticism we got was, are you modulating inflammation through intermittent fasting instead of right ventricular function? So then we went back to the drawing board, another 6 months of experiments, and then we used this model called Sugen hypoxia, which is a model that you put the rodents into a hypoxic chamber for about 3 weeks. They develop pulmonary hypertension by vasoconstriction, the same process we have when we have hypoxia. And then they develop pulmonary hypertension, and then after 3 weeks was the same process, and we used those 2 models accordingly. The one thing, as we're talking about intermittent fasting, that I want to highlight in any intervention, I feel like the importance of an intervention is how powerful the intervention is and for how long you keep that intervention. So to give you a perspective, this intervention in humans would be similar to 3 to 5 days of fast. The metabolism in rodents is much higher than ours. And here we're doing half of their lives or their whole life. So just to compare that people do intermittent fasting every other day, or they do for a couple months out of the year or something like that, how much of impact we had on this intervention. To see if it worked, we wanted to have a signature of the fuel-related proteins, the fuel-burning proteins, how the organelles looking, in that case the mitochondria. We want to see the heart function with an echo, and then we did pressure volume loops to evaluate pulmonary pressures, right ventricular pressures, and others. The first question we had was, we proposed this mechanism was going to come through AMPK, which is that sensor of energy. So the first thing we need to do is to prove that we achieve the AMPK activation we're after. And here you can see on the motocrotaline and the Sugen hypoxia that they activated AMPK, both here. And then when you look on the blue compared to the red, that you have significantly higher activated AMPK on both groups compared to the ad lib, the rodents that could eat. So now that we proved that the sensor that we're looking as a surrogate was activated. Now we want to see what happened to the fuel that we want to burn, and that's fatty acid oxidation. So we look at all the proteins that we know since the 1930s, they go down as the heart failure develops to see if they were spared. And this is what you have here on this hierarchical cluster. You have the ad lib in blue, which is pretty much just showing significant reduction of those proteins that control the fuel source, fatty acid oxidation. And when we look at the control and the intermittent fasting, you can see there's a significant reduction in that maladaptation into fatty acid oxidation, meaning you have more fuel available and more fuel ready for the cells to use. So now that we have more fuel, the question is, can we burn that fuel? And what we look next in this study was the mitochondria. Mitochondria organelle that burns the fuel. One of the main ways mitochondria burns the fuel is by having a lot of surface area. The surface area, when we look at the mitochondria, is cristae. The more cristae you have, the more surface area you're going to have to exchange electrons to create more energy. As heart failure develops, or any chronic disease for that matter, you start losing cristae. The mitochondria change shape and become a lot less efficient. So that's the one thing we want to look at here, which was the cristae score. So we look on the microscope and we counted every single mitochondria in the heart tissues of those rodents. And then what we saw is for the motocrotaline and the Sugen hypoxia, the cristae score with intermittent fasting was much higher compared to the ad lib, meaning you had more surface area within those mitochondria, either because they didn't get damaged in the first place or because we had mitochondria biogenesis and you had newer mitochondria which were healthier and able to perform electrical exchanges. So that's how the mitochondria looked. The next question was, can we prove that they're doing more electrical changes to create energy? So then we look at the electrical transport chain proteins expression in the mitochondria from the intermittent fasting groups and the ad lib. And what we've shown is this, where the most of the energy is generated, which is complexes 1, 2, 4, there was a significant increase in those complex in the intermittent fasting group compared to the ad lib. So at this point, we prove our energy concept, so we're ready to go, but we got this back, which was within the signature that we got for proteomics. We saw a very strong negative signal that was associated with right ventricular failure, meaning the inhibition of that pathway, whatever that was, protected the right ventricle. So then we didn't even know about this ACL4, and we went looking for, and then it has been shown in several other disease states, and that's true for ferroptosis. The heart we saw, and usually ischemic reperfusion injury, and we might heard from liver cancer and renal cancer this term ferroptosis. So that's what we found, this very strong signal that kind of came without us looking for it and without us knowing that was going to be a huge impact. This was the first paper published, we got this in JACC-BTS, that shown that inhibition of ferroptosis led to improvement in right ventricular function. The end result here, which was our question about the fuel, burning the fuel, can we do more mechanical work? That's what we care in the end of the day. And that's what we've shown here on RV2PA coupling, significant improvement with intermittent fasting by measures on echo, significant improvement. And that held true for the motocrotaline model as well as the Sugen hypoxia model. So summary of this work is that our intervention, intermittent fasting, activated the sensor with a lot of downstream effects, which is the AMPK. That led to improvement in the fuel source and the ability to use the fuel, in this case, the cristae in the mitochondria, that reduce the fuel from being stuck in the cell and getting oxidized and leading to this ferroptosis pathway. That improved the architecture of the cell, and the end result of all of those changes was improvement in right ventricular function. So we completed that work, and now we wanted to understand this ferroptosis cell death a little bit more. So ferroptosis is non-apoptotic form of cell death. To simplify is you have fat coming in, free fatty acids that you need for energy, and you're asking for more to come in. They get stuck. They start to get oxidized, and they generate this lipid reactive species. This reacts with iron that's bringing the oxygen on the cells. That's the carrier. They come together, and they generated this very, very inflammatory mode of cell death. Huge complement deposit, huge interleukin activation through this mode of cell death. And again, like I was talking about, this are multiple ways, but in the heart, that's just to think about how much inflammation we have in injury reperfusion, which we see often, which is in our MI patients, and we're trying to do a lot about it. And this ferroptosis works or happens in a lot of the organs. So based on that, we wanted to evaluate this disease process, which is pulmonary hypertension, very pro-inflammatory, and also effects on the heart. So we set out to prove the principle one. We saw this signal we were not targeting, came through the fasting. We don't know how intense that was. Can we replicate those findings? So what we did was we're going to activate ferroptosis. Can we create pulmonary hypertension by have a gene model of the same ACL4 that we saw inhibited? That was the first thing we did. The second thing we did was what happens with this? And then the second thing was, what if we inhibit ferroptosis? Can we have the reverse effect when we target ferroptosis only, as opposed to using a pleiotropic effect, which is fasting? So that was our study design. And first question was inflammation. Pulmonary hypertension, very inflammatory disease. Ferroptotic mode of cell death, very inflammatory. So we wanted to quantify on this ACL4 how much complement deposit there was. So this is in purple here, and you can see that on our genetic model of ferroptosis, there was a significant increase in complement deposit compared to the baseline. And here you can see when we quantify across how much more complement deposit, meaning how much more inflammation there was. When we reverse the course, which was, let's inhibit now and see what kind of effect we had. So for that, we use the control here. Similar thing, the purple is going to be the complement deposit or inflammation in other words. We use the same motocrotaline model, no ferroptosis, just a motocrotaline model, nothing done to it. And you can see that highly inflammatory model as well, a lot of this complement deposit. And then this same motocrotaline model, now with ferrostatin 1, which is an inhibitor of ferroptosis, and we see a lot lower complement deposit, meaning a lot less inflammation on this very inflammatory model. So what does that mean? Pulmonary hypertension, as you know, you start having changes in the vasculature from inflammation that leads to thickening of those vessels and reduction of the compliance. Those vessels become very, very stiff. So what we wanted to see is how do the vessels look when you have this genetic model of ferroptosis. And what we see here is significant increase in medial wall thickness in this proferroptotic model. When we do inhibit on our motochronoline. So see the motochronoline, how much sclerosing agent and how much fibrosis and thickening of the media you have compared to the control. See how thin those walls are. The ferrostatin 1 inhibition, you had a much lower degree of increase in the thickness compared. The end result, like we talked about it, we want to know is what about the right ventricle? And that is right ventricular function. So when we evaluate ferrostatin 1 inhibitor, inhibiting ferroptosis, we saw improving in coupling, right ventricular to PA coupling. We saw improvement in markers of right ventricular function. And that showed that our intervention separately targeted had benefit in the pulmonary circulation and also in the right ventricle. At that point, it was not clear to us if, because we improved the pulmonary circulation, we had less pulmonary disease on our model. That's why the right ventricle did better, or it was because the right ventricle did better, period, because we improved the bioenergetics. But that was what we saw, improvement in the right ventricle. This is an animal model. This is an intervention. The biggest question we had was, what do we know about humans? How does this impact humans? So for this, we collaborated with Evan Britton from Vanderbilt. We have long track record of publications together. And we looked at the Vanderbilt Biobank database using SNPs. What we found was there are 6 pro-ferroptotic genes. And the patients with pulmonary hypertension, they are carriers of those genes, have a much more severe pulmonary hypertension course. So they have higher right ventricular systolic pressure, they have higher mean pulmonary arterial pressure, and they also have higher pulmonary vascular resistance. So that was human data that we had. Based on this human data, showing that ferroptosis increases pulmonary resistance, inhibition reduces and improves right ventricular function, we wanted to prove that in humans. Because it's not so easy to get there, we need to have an intermediate step. And that was to find a model that was most close to humans. So we decided to do a model that was similar to human physiology. We chose a large animal model of pig, and we did a pulmonary artery bending. This is an MRI of the PA here, so you can see the band right there. And you can see the aorta to the side, just to give you an idea of how much of predilation you have on the PA and also post compared to the aorta. When we look at that flow, this is how it looks. So this is an MRI again, and this is right at the area that we put in the ring surgically, how much of tightening and flow, and you have that jet here, very significant. We had about 95% stenosis generated on area by area calculation across our study. To show that the model worked, we did MRI to look at the right ventricular function, and this is a RV two-chamber view, so you have your SVC coming here, right atria, your IVC is not here on the plane, but you can see on the control, or a normal pig, a significant contraction on your RV two-chamber and normal size. On our banded animals, the model we're creating, on the other hand, show very dilated right ventricle, tricuspid regurgitation. If you look here, the free wall barely moves. The only thing that does a little bit is the base of the right ventricle, this apse. So very significant right ventricular dysfunction. When we look at a 4-chamber here, this is our control, and this is our model. You can see the size of this right ventricle, 3-4 times bigger than the control. Atria got dilated as well here, as you can see. And then you had a shift, as you can see here, the shift on the interatrial septum, it was towards the right. But here on our model, pressure overload on the right atrial chamber, shifting the septum towards the left in this case. Here at the bottom, you have this flow acceleration here showing the banded area on the PA. So we published this showing that, one, our model works. We can create right ventricular failure in the large animal model. And we have increase in right ventricular mass, and we also have increase in reduction in right ventricular function, and that was done by Prince with our group at the university. The second question we had was, how similar is this model in signature compared to humans? So one, we've shown the model has a very significant change compared to the control in the metabolism. As we talked about, as you develop heart failure, you change your metabolism. We're proof that our model does change metabolism. That's what we're seeing here in blue and the opposite. And then we compared to patients with decompensated right ventricular failure, and we saw they had the most similar metabolic signature to the large animal model. So now that we had this model, we set out to do this last phase of the research was target therapy for the right ventricle. So our goal was, we had this lipid that we need to use for fuel. We need to burn it, but it gets stuck and oxidizes, creates this ferroptosis. We target with this ferrostatin inhibitor. We want to see what happened to the burning cells, mitochondria, with our large animal model using cardiac MRI to look for the function in PV loops and microscopy. So this is the paper we just published last month in the Blue Journal. And the first thing we shown was that ferroptosis inhibition prevents the abnormalities in the right ventricle fuel source, which is you have more oxyphosphorylation proteins in the ferrostatin 1 group, meaning you're able to burn more fuel. And the second factor that we saw that was very interesting is right ventricular triglycerides, meaning accumulation of intracardiac fat deposit, steatosis in a way, was much lower in the ferrostatin 1. Here in blue, you can see significant, here in placebo, sorry, and then ferrostatin 1 significantly reduced with this dark color. This is the MRI results, and that's what we're going to have until the end, is just showing our phenotype with our intervention. So here you have a two-chamber dilated right ventricle. You have your SVC, your IVC coming in. It's a nearly bicaval view on MRI. We don't use for clinical, but for research, it's just a nice view. And then you can see how much TR you have here, and more interestingly, how the TR is directed towards the inferior vena cava here, creating a lot of turbulent flow there. We look at our intervention, which is ferrostatin 1, in a model that has no pulmonary arterial disease. We bend it. There's no vasculature disease to be the reason for our ferrostatin 1 to work or not, like our rat model. And we show significant improve in the coupling. You have, as you notice here, increase in thickness, hypertrophy of the right ventricle in a compensatory mechanism. You have a much lower cavity size and improving contractility of the right ventricle. When we look on short axis, we see the right ventricle in the control models severely enlarged, severe hypokinesis, a very impressive septal bounce with septal compression of the left ventricle. Outer geometry, as you can see here, the longitudinal anterior to inferior is much longer than septal to lateral as you're changing the geometry of the left ventricle. And when you compare to our intervention, you still have evidence of increase in PA pressures by this septal bounce, but a lot less pronounced. The geometry of the left ventricle somewhat preserved. And then you have this compensatory hypertrophy, making this more of a coupled with a PA and the pressures. Our model also shows something that we see in our patients very often, which is you start with one problem, whatever that is, AFib, pulmonary hypertension, RV failure, and then you get into the second problem, which is as your annular dilation happens, you get a volume overload hit on top of your pressure overload hit. And that's what we saw on the model. As dilation happened, they develop severe tricuspid regurgitation. And you can see here on short axis MRI on FOSS with a valve that you have a very significant central jet just from being stretched out from the volume dilation. So then we had a two hit phenomena, unfortunately for the control, which was pressure and then secondary volume overload. And that's the one thing that also for statin inhibition prevented, which was we had mild to moderate TR in the first statin inhibition group. And then here you can see on short axis at the level of tricuspid valve, much smaller annulus and not significant tricuspid regurgitation as well. And then you can see what happened to the atria. Look at the atria, it's barely contracting. It can very dilated as compared to the atria on the first statin. So in summary, we proposed a ferroptosis inhibition as a target therapy for the right ventricle for the benefits that we've shown in the pulmonary vasculature, for the benefits regarding inflammation, as we've shown the complement deposit, and for the benefits directly into the right ventricle, as shown in this model here, when we're comparing a 4-chamber control to a 4-chamber for statin 1 inhibition targeted. This is our results. So we've shown about an 11% increase in right ventricular ejection fraction from severe, moderate severe to mild. We also shown a significant decrease in dilation of the right ventricle, and more importantly to our EP colleagues, but something that we're learning more about it, which is the right atrium, significant improvement in right atrial function as well. Maybe TR, maybe because of compliance, maybe a combination of factors, but we did see this change as well. So we do believe that for all of these benefits for statin 1, it is a potential target that should be further investigated in patients with pulmonary arterial hypertension for this dual benefit, but also in patients with right ventricular failure for this single benefit that we're showing in this paper. With that, I would like to thank you all for this opportunity, and I couldn't have done, this is about 4 years worth of work, and I couldn't have done with this team, so they're very talented, and they helped me throughout, and they're gonna be the future of medicine in a way. So what we have here is Neil. He worked with us on the Ferroptosis projects for about 2 years, and now he's going to PA school. He just got accepted. And then Jenna is a PhD. She continues to work with us. Prince, he's my mentor, and he runs the lab at the University of Minnesota. Rashmi, very young and brilliant. She's wanting to go into med school. Sasha Prisco, she works with Kurt and joined the university as PhD, MD PhD program. Anna Park, she also worked with us for several years and went to med school last year. And then we have Jacob here on the back. He's going into vet school. And then we have Madeline Blake, which is brilliant. She had addiction issues with her family, but she was very resilient and then created a non-for-profit organization for students that have issues with addiction, and she wants to go into MD, PhD school. So this group made it possible, so I'd like to thank you them all.
Phenomenal, Felipe. That's great. Amazing work. Amazing work. Questions? Jay?
Thanks for that great talk. So how does, I was just wondering if you could compare and contrast, we have a strong interest in intramyocardial hemorrhage following ischemia reperfusion injury. Is Is it, and there's also a lot of lipid peroxidation that goes on with that, and I'm just wondering, compare and contrast your model on the right ventricle. Is that is similar biology going on?
So that's a great question. One way of testing would be using the LED ligation or balloon occlusion for us to see how much of the ferroptosis signal we have. But I do believe that with ferroptosis and the lipid peroxidation that you have with ischemia reperfusion, you're going to have a very, very inflammatory response that maybe the ferrostatin one would also be something that to benefit those patients.
Okay. And then we're going to be starting a clinical trial using an iron chelator instead of ferrostatin one that you're using. And I'm wondering, have you tried an iron chelator to see what the effects are on your model?
No. So what we learned this was about 2 years ago when we even learned about this pathway of ferroptosis. And that's when we did two things. Proof of principle, does it cause pulmonary hypertension, and how can we inhibit and prevent it? And that was through the genetic model using the ACLL4. Chelator would be a possibility. What we feel like would be more of a target, because our intention is to develop a treatment for biventricular failure. And then we feel like a target therapy, which we'll be using for both, would be a more powerful approach. But that definitely is a bigger player. The iron's a big player in this whole process. Have you guys tried to use anything related to ferroptosis inhibition in that model? No?
Potential another pathway to
Absolutely.
target. Yeah.
Yeah.
Thanks, Felipe. Were you on the MRIs you were showing on the pig model? Were you showing treatment effect? Were you showing same animal and then before and after treatment, or are those two different animals?
Well, that's a great question. So we had 3 groups. So we had the control, which we did nothing to it. They just got a sham operation. And then we had the pulmonary arterial bandit, which is this animal, 5 weeks in. And then we had our model, which got the surgery at the same time, but got treatment right away after surgery. And
treatment began immediately
after surgery.
Treatment.
So this here, no treatment. And then on the left side of the screen is treatment for about 5 weeks from the surgical date.
Just thinking about it in terms of potential human application, and when we treat patients with pulmonary arterial hypertension, we treat the pulmonary vasculature as best as we know how and hope they don't develop right ventricular failure. Some do. And the reason I'm asking the question is what would happen if the treatment began after full-on RV failure occurred? Would that be, is it reversible?
Yeah, that's a great question, and that's something that we talked about it too. The issue it comes in the end of the day is logistics and cost. So for us to prove that these animals, so first of all, these animals are very sick. We had several of them that died just straight up PA arrest from being this sick. So we have a 95% banding of area on the PA. The way we would do when we talked about it was we have to have this model and then do imaging on them until we prove that the model developed this RV dilation, RV failure, and then start treatment. So that's definitely something we're going to consider in the future, but the big issue is going to be a cost to keep doing echo on them until we see how many weeks it took for them to develop.
I actually have about 1,000 questions. Is the ferrostatin, is the ferroptosis inhibitor a very toxic medication?
Yeah, excellent question as well. And that's one of the challenges.
The question is there even any potential human application?
Well, there is. So regarding cancer treatment, more specifically, there is treatment using ferroptosis inhibition or modulation in a way. And there's also studies showing that doxorubicin, which is lipid in a way, oxidation, things like that, induce cardiomyopathy. When you use ferrostatin inhibition, you protect the heart from the cardiomyopathy. But the major issue is not as much as toxicity. The 2 major challenges we're facing is, one, solubility. This is an injection that we have to do every day on these animals, and that will not be something that will be applicable to people at the doses we're using. And then the second one is that it works in all cell processes. So we don't have a way yet to have only on the pulmonary vascular ferroptosis inhibition and the right ventricle ferroptosis inhibition. And that's going to be the major issue to overcome, which is this is a normal process, this is a healthy process, it just gets activated too much when you have disease. And how can we have it that it doesn't affect the whole body? That's the major challenge. Not toxic, but affecting the whole body over long term, we don't know.
First of all, thank you very much for this excellent presentation. So my question is, so we know in the pulmonary hypertension patients, secondary hematopoiesis is a big problem. So how does the ferroptosis inhibitor affect the hematologic parameters? So like because for example, in Eisenmenger patients, like we are trying to increase their iron level instead of decreasing some potential problems. So I just wanted to ask, like, have you ever looked at the hematologic problem, like hematologic parameters?
Yeah, excellent question. Thanks for bringing that factor up. So 2 points that I'll make. One is our animal model here is about 8 weeks total. They're 3 weeks in and then 5 more weeks or so after we develop. So we don't have long-term data to evaluate cells. But the major point that I want to make is, and it's not only our group, there's several groups shown that ferroptosis is very pro-inflammatory. And then we know that anemia of chronic disease is a pro-inflammatory condition. We know the inhibition of ferroptosis significantly reduces inflammation. So if I were to speculate, I'll say that process from the inflammation reduction would be improved on the hematopoiesis. But we don't have data from our group about that.
Any questions from online or?
No questions on.
Okay. Felipe, that was fantastic. Thank you again. And